Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 6.7, mutations. So lots to talk about today, but we're going to start by discussing point mutations and then we'll move on to errors in DNA replication and DNA mechanisms as well as mitosis, and lastly talking about horizontal gene transfer. And our suggested skill visual representations 2C explain how biological models relate to larger principles, concepts, processes, systems, or theories. So as we've discussed a few times before in the course, a mutation is a random alteration in a DNA sequence. And it can affect changes in the amount and type of a protein produced, because if the nucleotide sequence is changed, then different proteins will be produced once that section of DNA is going to be expressed through transcription and translation. And so first we're going to focus on point mutations, where there's an alteration of just one nucleotide base pair. One is substituted for another, or we have an insertion/deletion where there's an addition or loss of a nucleotide base pair. And this causes a frameshift mutation because it shifts over the codon grouping, and so it's a mutation that interferes with the codon grouping that that groups three nucleotides into one codon into one protein. So if you look over here, our normal sequence of DNA, ATG, CAT, and then TGC, those are three codons because we have our three nucleotides in each codon. And so then going through the transcription and translation process here, for this sequence, first we start with mRNA, so UAC, and I'm just doing base pairs here, then GUA, then ACG, and then going to the translation process with tRNA, AUG, CAU, and UGC. So you can see your AUG, that's going to be your start codon, and then when we look into our codon chart to see what amino acids this is going to code for, we start with the methionine as a start codon. Then, so we have MET for AUG, then let's look at CAU, CA over here, and then U, that's going to have HIS, and then with UGC, UGC, that's going to be cis. So these are the different amino acids that we can see on the codon chart from this coding. However, if you have an insertion, if there's a thymine nucleotide inserted right over here after this T, then the reading frame has been changed because this is now the codon. Instead of ATG, it is now ATT. Then the G becomes, the guanine becomes part of the next codon. This thymine becomes, and so all of these codons are screwed up, so there's going to be different proteins that are then produced. And nonsense mutations, when this happens, can cause a premature stop codon. So a stop codon means there's going to be either UAA, UAG, or UGA when you do the translation. And so nonsense mutation can cause a premature stop codon before the amino acid sequence and the polypeptide is complete. But there are also silent mutations where there's no effect on the resulting protein. And that's part of why this chart, as you can see here, is so redundant. For instance, for valine, there are 4 different options that would code for the same amino acid. So even if the last letter was changed due to a mutation, the same amino acid would be coded for. And so this codon chart is redundant because sometimes we can get these silent mutations where even though a mutation occurs, there's no effect on the resulting protein because the DNA is still going to code for the same protein. So mutations are caused both by substances that interact with DNA, known as mutagens, such as radiation, which can cause mutations, but also just errors in DNA replication and DNA repair, errors in the cell cycle during mitosis and meiosis. Those can all cause mutations. And then any alteration in the chromosome structure can lead to genetic disorders. And today we're going to be looking at non-disjunction. That's when changes in chromosome number lead to changes in the phenotype and genetic disorders. And non-disjunction is when chromosomes do not separate properly during cell division. And the result is that the cells are not identical, as you can see with the amount of genetic information they have. And this can also occur in meiosis where the gametes do not have, they either have too much or not enough genetic information there as well. And so if you get an extra chromosome within the cell, then that's known as aneuploidy, and we have something like Down syndrome as a result of having that extra chromosome. Now, now we have to talk about horizontal gene acquisition. And so we're focusing on prokaryotic gene acquisition, and this is how the prokaryotic genome can change in addition to mutations. So the first one is known as transformation. That's when DNA is taken in the form of a plasmid and added to the genome. And the plasmid is just a small ring of extra chromosomal DNA. We discussed this in 6.1, but it's just a ring that's not part of the regular genome of the bacteria. It's just another piece of DNA, but then it can be integrated into the bacterial DNA, and then those genes will be expressed by the bacteria. Now there's also transduction, where viral DNA is taken in and added to the genome, and even related viruses can recombine DNA if they infect the same host cell. So within transduction, the virus, in this case we're looking at a bacteriophage, so that's a virus that attacks bacteria. This transferred DNA will be added into the genome, and then that's how new phages are going to be created. And a virus is an infectious agent that replicates only inside the living cells of an organism. So in order to replicate, transduction must occur. We also have conjugation, where DNA is transferred through cell-cell connection, when it then can be incorporated into the genome of the other cell. And lastly, we have transposition, which is the movement of DNA segments between DNA molecules. So within the same DNA molecule, segments can move around, changing gene expression. And kind of as a whole, mutations can be beneficial, detrimental, or neutral. They're not always a bad thing, and it just depends on the environment, because if the mutation actually allows an organism to survive better in an environment, then it's going to be passed down. It's beneficial. So not all mutations are bad. And when we look at a mutation like sickle cell anemia, which turns blood cells into a sickle shape, this was a mutation that eventually became part of the genome and is a trait that can be coded for. And the reason it's still being passed down between generations is because when you're heterozygous for sickle cell anemia, then you have a slightly stronger resistance to malaria. So it's passed down because it's still useful to an organism surviving in an environment where there is a lot of malaria infection. And it's also key to remember that mutations are the primary source of genetic variation, and that's going to come in play when we talk about Unit 7. But for now, just know the different kinds of mutations, what causes them, horizontal gene transfer, and how that contributes to genetic variation within prokaryotes. And remember that not all mutations are bad.